Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome to your channel, The Anatomy Canvas. In today's class of the neuroanatomy, we are going to discuss the cerebellum. So the learning objective for the today's class are to describe external features of the cerebellum, to discuss the internal features in detail, and to discuss different neural circuits between deep cortical nuclei and with different tracts. So we will discuss all of the things related to cerebellum in this lecture. The gross anatomically, the cerebellum occupies the posterior cranial fossa and separated from the occipital lobe of the cerebral hemispheres by the tentorium cerebelli. The largest part of it is largest part of the hindbrain, and it is dorsal to bone and medulla, from which it is separated by the fourth ventricle. So in this diagram, you can say this is the superior surface of the cerebellum, this is the inferior surface of cerebellum, and this one is the cross section of the cerebellum. This one is the anterior lobe, the posterior lobe, and this in blue color is the flocculonodular lobe of the cerebellum. We will discuss all of these features in detail now. So three bilaterally paired cerebral peduncles which attach the cerebellum to the brainstem. The superior, middle and inferior cerebellar peduncle. The superior attaches the cerebellum to the midbrain. The middle attaches the cerebellum to the pons, and inferior attaches the cerebellum to the medulla oblongata. So it is the only region of the CNS that spans the midline without interruption. In this diagram, you can also see that it is the area where it spans the midline, and it spans the midline without any interruption. It occupies almost 10% of the total brain volume, but it has half of all the neurons in the brain. The surface is deeply inconvoluted in the form of the folia to accommodate these large number of the neurons present in the cerebellum. It is embryologically derived from the ectodermal thickening about the cephalic border of the fourth ventricle, which area is called the rhombic limb. That is why it is also called the rhomencephalon. Main function, first we will have to see the main function, then it will be easy for us to understand the different neural circuits of the cerebellum. So main function of it is the maintenance of the posture, the maintenance of the muscle tone, and the coordination of the voluntary motor control. These are three main primary function of cerebellum. So in this diagram you can see these are the different relationship of the cerebellum. Here the occipital lobe of the cerebral cortex is is the tentorium cerebelli. Here is the pons and the medulla and the midbrain, and it is present posterior to this brainstem, and in between the brainstem and the cerebellum you can see the cavity of the fourth ventricle. So the external feature of the cerebellum, basically it consists of three parts: the two surfaces, the two notches, and three very well defined fissures. Some books even describe the 5 fissures as well. So the three parts of the cerebellum are the two cerebral hemisphere, and in the center is a worm like area called the vermis. So the two cerebellar hemisphere and the central vermis, these are the three parts of the cerebellum. Then the surfaces is the superior surface and the inferior surface, and the two surfaces are separated from each other by the horizontal fissure. So the external feature, then the notches, we are going to see the anterior and the posterior notch. You can see here the anterior notch which is present, and the posterior cerebellar notch. This one is the superior surface and this one is the inferior surface. Again you can see the posterior notch over here and the anterior notch. Then the three main fissures, the three well defined fissure, the horizontal fissure, we divide the surface into superior and inferior surfaces. Then the fissura prima, or the primary fissure, which is present over here. This is again a very well defined fissure which separates it into anterior and the posterior lobes. Then the third fissure, the posterolateral fissure, which separates the flocculonodular lobe from rest of the cerebellum. So in this diagram you can also see the center part is the uvula. On the each side you can see the cerebral hemisphere, and these are the different lobes, and this fissure is the posterolateral fissure, which is separating the flocculonodular lobe from rest of the cerebellum. So these are the different features, the fissures which are present. This one is the horizontal fissure, which is present over here, and it separates the superior surface from the inferior surface. Now this is again the model, and you can see on this structure, you can see this is the cross section of the cerebellum, in which you can see in superior surface is related to the cerebral cortex, and anteriorly it is related to the brain stem, and the cavity between the two is the fourth ventricle. It is attaches to the brain stem by means of the three peduncles, the superior, the middle, and the inferior cerebellar peduncles. Some of the books also describe the external features, that there are the five fissures which are present in the cerebellum. Already most of the books describe the primary fissure, the horizontal fissure, and the posterolateral fissure, but some of the books also describe two other fissure, that is posterior, superior, and the prepyramidal. So in this diagram you can see this one is the primary fissure, the main fissure, which separates the anterior lobe from the posterior lobe. Then you can see the posterior superior fissure, which is present over here. You can see this one is the posterior superior fissure, which separates these two lobes of the posterior lobe, these two lobules of the posterior lobe from each other. This is the postero superior fissure. Then third is the prepyramidal fissure. Here you can see this area is the pyramid, and this is the prepyramidal fissure which is present. So this the anterior to this primary fissure is the anterior lobe, and then this whole is the posterior lobe, and this in purplish color area is the flocculonodular lobe. So again the anterior lobe is divided into three parts. This one in the pink color is the lingula. In the blue color you can see the central, and in the yellow color you can see the culmen. Similarly on the superior surface you can see the central and the culmen, and inferior surface also you can see the lingula as well, and the central as well. Then the posterior surface, the first in the posterior surface, this one is the simple lobule. Again you can see over here, this is the simple lobule, then is the folium, and then is the tuber. So these three, the simple lobule, the folium, and the tuber. Again in this diagram you can see the simple lobule, the folium, and the part of the tuber. This one is the superior surface, and this one is the inferior surface, in which you can see large part of the tuber as well. Then the third next part to this is the pyramid, and between the pyramid and the tuber you can see the prepyramidal sulcus, which is present over here, and next to the pyramid is the uvula, and then in the purple color you can see the flocculonodular lobe. Similarly over here you can see this one area is the pyramid, and this is the uvula, and then you can see the flocculonodular lobe. So these are the different lobules which are present in anterior and posterior part of the cerebellum. Again I repeat, this one is the lingula, then this one is the central, and then this one is the culmen. Similarly here this smaller is the lingula, this in blue color is the central, and this in the culmen. Then is the primary fissure, which separates the culmen from the simple lobule. Then simple lobule is the part of the posterior lobe, and next to the simple lobule is the folium, and then is the tuber in the green color. Next to the tuber is the pyramid, and between these two is the prepyramidal sulcus. Then is the lingula, uvula, and next to uvula is the nodule, which is part of the flocculonodular lobe. And this here you can see the posterolateral fissure, which separates the flocculonodular lobe from the posterior lobe. So these are the different parts, the different notches, the different fissures, the different lobes of the cerebellum and its grass features. Now the functional subdivision of the cerebellum. The functionally cerebellum is subdivided into three main parts. The one is the spinocerebellum, the other is the pontocerebellum, and the next is the flocculonodular lobe. The spinocerebellum is also called the paleocerebellum, and this spinocerebellum mainly consists of the anterior lobe with a part of the simple lobule and the pyramid as well. Here you can see this one is the simple lobule, this one is the pyramid, and this is the anterior lobe. These three parts constitute the spinocerebellum, which is related to the main recipient of spinal and trigeminal cerebellar efferent fibers. Next is the pontocerebellum. It is also called the neocerebellum. This pontocerebellum is very well developed in human beings. It carries the cerebro-pontocerebellar pathway, and it dominates in the folium, in the area of the tuber, and in the area of the uvula, and throughout the entire hemisphere. So this in the blue color is the pontocerebellum, and this pontocerebellum is the neocerebellar. It receives the efferents from the it receives the efferent from the spinal cord. So these are the neocerebellum. The vestibular cerebellum is oldest of all, and it is formed by the flocculonodular lobe. And then the efferent of the cerebellum, the efferent projection of the cerebellar hemisphere through dentate, emboliform, and globus nuclei to thalamus, and then to the cerebral cortex. So these are the three deep nuclei which are present, four deep nuclei which are present: the dentate nucleus, the emboliform nucleus, the globose nucleus, and the vestigial. The very simple to memorize this is: don't eat greasy food. Don't for the dentate nucleus, eat for emboliform nucleus, greasy for the globoses, and the food for the vestigial. So these are the four deep nuclei which are present in the substance of the cerebellum, and functionally it is divided into spinocerebellum, the pontocerebellum, and the vestibulocerebellum. Then the cerebellar internal structure, you can very well see these are the folia, and then the internally here you can see the outer is the cortex, and inner is the white matter which is present. And in this inner white matter you can see the deep nuclei which are present, which I have already told you that these are the dentate nucleus, the emboliform nucleus, the globose nucleus, and then the vestigial nucleus. So this is the internal structure. Little bit about its histology. The cortical gray matter is divided into three parts. The outer is the molecular layer, then is the Purkinje cell layer, and then is the granular layer. So most external molecular layer mainly a synaptic layer with few pericardia and many unmyelinated nerve fibers. The types of the cells which are present in this outer molecular layer are these are the basket cells and the stellate cells which are present in this molecular layer. Then here you can see these are the stellate cells which are present, and the basket cells which are present deep in the molecular layer. The stellate cells are basically star-shaped cells. They have many short dendrites and fine unmyelinated axons. Axon makes synapses with the dendrites of the Purkinje fiber. So this is the Purkinje fiber, Purkinje cell, and its dendrites are present in the molecular layer, and these dendrites have a connection with the axons of the stellate cells, and these have a negative feedback effect on the dendrite. These are inhibitory in nature and release the GABA. Similarly, the basket cells, nearly 15 to 20 basket cells arrange themselves around a single around the cell body of the single Purkinje cells, and they form a basket-like structure. That's why also called the basket cells. They have many dendrites, and they have the unmyelinated axon which runs horizontally and give terminal arborization around the body of the Purkinje cell. Here again you can see these are the axons of the Purkinje cell. These are the axons of the stellate cell. Both these two types of the cells are inhibitory in nature, and their neurotransmitter to the Purkinje cell is the GABA, which is the gamma amino butyric acid. Then the middle layer are the Purkinje cell layer. Here you can see this is a uniform arrangement of the Purkinje cells. These Purkinje cells, the largest and most numerous neurons present in the central nervous system. They are arranged in a single layer of the neurons with pericardia lying between molecular and the granular layer. So their pericardia, their cell body lying between molecular and the granular layer. They have highly branched dendritic tree. You can see over here highly branched dendritic tree, and axons of these travel into the Purkinje layer and to the deep cerebellar nuclei, and also they give some collateral branches to the Golgi type 2 cells. So inner is the granular layer. This inner granular layer again having the two types of the cell. The one is the granule cell and other is the Golgi cell. The granule cell is the smallest neuron present in the body, about a diameter of 5 micrometer. Neurons are multipolar with 3 to 6 dendrites, and the other types of the cells which are present over here are the Golgi type 2 cells. So these are the different neural connections. Very interesting diagram to understand. First we will start from here. This is the inferior olivary nucleus and their fibers going and enter into the cerebellum as the climbing fibers. They give first a branch to the deep cerebellar nuclei, and here they secrete the aspartate as the neurotransmitter, and again these fibers ascend through the white matter through the granular layer into the Purkinje cell layer, and then these climbing fibers stimulate the Purkinje cell, and again in the neurotransmitter over here is the aspartate. In turn, these stimulated Purkinje cells, their axons goes to the deep cerebellar nuclei, and these axons are inhibitory in nature, and they over here release the GABA. So the climbing fibers from the inferior olivary nucleus, they stimulate both the deep cerebellar nuclei and also the Purkinje cell nuclei, and Purkinje cell nuclei in turn inhibit the deep cerebellar nuclei. So these are the climbing fibers. Next are the mossy fibers which are present. All other sensory input to the cerebellum is the mossy fiber, that is to the spinal cord, to the brainstem nucleus, and these mossy fibers, when they ascend, they give first a collateral branch. This is again excitatory neurotransmitter which is present over here. They stimulate the deep cerebellar nuclei, and then these mossy fibers stimulates the granular cells. These granular cells are present in the granular cell layer, and these granular cells these are the cells. First, these are the cells with Golgi type 2 cells which are stimulated by a branch of the collateral branch of the Purkinje cells, and upon stimulation these Golgi type 2 cells inhibit the granule cell. So must remember, these mossy fibers are stimulating the granule cells, while these Golgi type 2 fibers are inhibitory to the granule cells. The axon of the granule cells then travel into the molecular layer where it divides into a T-shaped manner, and this axon also has the connection with the Golgi type 2 cell, with the basket cells, with the stellate cells, and also with the cell body of the Purkinje cell. To all of the cell, the axon is a stimulatory in nature, while the basket cell and the stellate cells, when they are stimulated by the granule cells, they again have the axosomatic connection with the Purkinje cells, where they are inhibitory in nature. So please remember, the climbing fiber are excitatory both to deep cerebellar nuclei and Purkinje cell. Similarly, the mossy fibers are excitatory both to the granule cells and to the deep cerebellar nuclei. The granule cell is excitatory to the Golgi cell, to the basket cell, to the stellate cell, and to the Purkinje cell. But the stellate and basket cells and Purkinje cells are inhibitory in nature, and these two cells inhibit the Purkinje cell, and upon stimulation these Purkinje cells inhibit the deep cerebellar nuclei and also give a collateral branch to the Golgi type 2 cell, which upon stimulation inhibit the granule cell. So very important, very interesting connections which are present in the cerebellum. Again, a structure which is seen histologically also on the electron microscopic picture is the glomerulus, which is present in the granular layer of the cerebellar cortex. So this one is the mossy fibers, and these are the rosettes of the mossy fibers. These in pink color are the granule cell dendrites, and these in yellow color are the dendrites and the axonal process of the Golgi type 2 cell. So connection of the axonal and the dendritic process of the Golgi type 2 cells and the granule cells and the rosettes of mossy fibers, they make a structure in the granular cell which is called the glomerulus. Now the different pathways which are present in the cerebellum. The first is the spinocerebellar pathway. The spinocerebellar pathway have different components. They are the one which is present above the level of C8. That is the cunocerebellar pathway. This is present above the level of the cerebellum, and it starts from the accessory cuneate nucleus. Next is the posterior spinocerebellar pathway, which is present from the C8 to L2 or L3. Again, you can see this one is the posterior spinocerebellar pathway. This is again ipsilateral. The second order neurons are present in dorsal nucleus of the Clark, and they ascend to the deep cerebellar nuclei. Then third is the anterior spinocerebellar tract, which starts from the level of the L2 or 3 to the sacral levels and below. Their fibers, first order neurons are present in dorsal root ganglia, second order neurons are present in the spinal cord, and after relay they crosses to the opposite side and they ascend then the anterior white matter as the anterior spinocerebellar tract and into the cerebellum. They crosses to the up to the level of the superior cerebellar peduncle into the brainstem. They crosses to the opposite side and enter through superior cerebellar peduncle into the cerebellum. So this anterior cerebellar tract, it crosses twice, once in the cerebellum and next in the brainstem. So all of these tracts, the spinocerebellar tract, they are ipsilateral. Again, another tract which is present is the rostral spinocerebellar tract, which is present in the region where is enlargement of the spinal cord at the level of the brachial plexus, and it is considered as counterpart of the anterior cerebellar tract, but remember the difference is that it ascends ipsilaterally. So these are the different spinocerebellar tracts, and they are conveying the sensations to the cerebellum. The different connections, you can see these are the different lamina, the A, the B, C1, C2, C3 and D lamina. So A lamina is this one is the A lamina, which is attached to the vestigial nucleus. The fibers from to the A lamina which are coming to the vestigial nucleus. The fibers which are coming to the C1 and C2, these are the fibers which are coming from the inferior olivary nucleus from its different nuclei, and the fibers which are coming from the inferior olivary nucleus to C1 and C3, they relay in the emboliform nucleus, while fibers from the inferior olivary nucleus which is coming to the lamina C2, they are coming to the globose nucleus. So these are the different nuclei. This all area is the D area, and the fibers which are coming to from the posterior part of the inferior olivary principal nucleus of the inferior olivary complex, they come to the D area and they relay in the dentate nucleus. So these are the different efferent connections which are coming to the cerebellum cortex through the inferior olivary nuclear complex. Again, the efferent connection, the pontocerebellar fibers coming through the middle cerebellar peduncle. Here you can see these are the pontocerebellar, the efferent fibers, this arising from the cortex, coming through the internal capsule and then into the midbrain and into the pons, where they relay in the pontine nuclei, and then these pontocerebellar fibers then enter through the middle cerebellar peduncle into the cerebellum. So this tract is also called the cortico-pontocerebellar pathway, and it enters the cerebellum through the middle cerebellar peduncle. Then the efferent connection in the present in the inferior cerebellar peduncle are the dorsal spinocerebellar tract, the olivocerebellar, the vestibulocerebellar, and the reticulocerebellar. So these are the different tracts. Here you can see the vestibular nuclear complex is different part, the superior, the lateral, medial, and inferior part of vestibular nuclear complex, and its fibers are going to the B lamina of the cerebral cortex, which is then attached to the vestigial nucleus. So these are the different tracts. Now the efferent fibers which are coming to the cerebellum, which are going to from the cerebellum to different area, these are from the superior cerebellar peduncle are the dentatothalamic pathway and dentatorubro-thalamic pathway, and also the cerebellovestibular. So here you can see this is the superior cerebellar peduncle. Now remember the fibers which are going from the cerebellum to the cerebral cortex, they are not direct. First they relay into the thalamus. Either they go directly to the thalamic area, or first they relay in the red nucleus and then they go to the thalamus, and next to the thalamus in the VPL nucleus of the thalamus, and from here they go to the motor cortex. So must memorize that the fibers from the cerebellum do not directly go to the cerebral cortex. They have either dentatorubro-thalamic pathway or direct dentatothalamic pathway, which then goes to the cerebral cortex. Similarly, the cerebellovestibular fibers which go directly to the vestibular nuclear complex are also carried through the superior cerebellar peduncles. Fibers which are going through the inferior cerebellar peduncle are the cerebelloreticular fibers. Here you can see the nucleus reticular pontis, and these are the cerebelloreticular fibers which are coming to the reticular formation. Similarly, the cerebellovestibular fibers which are going to the vestibular nuclear complex are also going through the inferior cerebellar peduncle. So these two fibers, the cerebelloreticular fibers and the cerebellovestibular fibers, are going through the inferior cerebellar peduncle, while the fibers which are going to the cerebral cortex, they are going through the superior cerebellar peduncles. The efferent connection of the cerebellar hemisphere, again you can see this is the medial strip, and this one is the called the medial strip or the A zone, which projects to the rostral part of the vestigial nucleus. Here you can see this is the vestigial nucleus, and this is related to the A part of the A strip or the medial zone of the nucleus. These are the efferent connection which are present in the cerebellar hemisphere. Next is the B zone or the lateral slip, which projects to the lateral vestibular nuclear complex. Here you can see this is the lateral vestibular nuclear complex, and these fibers which are projecting to the vestibular nuclei, they are arising in the lateral strip or the B zone of the cerebellar hemisphere, in the part from the part of the vermis. This is basically part of the vermis because this is the flocculonodular lobe, which is related to the vestibular system. So A zone which is attached to the vestigial nucleus, and B zone which is attached to the lateral vestibular nucleus. The B zone does not continue beyond the simple lobule. Here you can see the different lobules. This is the primary fissure and this is the simple lobule. So you can see the B zone is only present in the anterior part and in the simple lobule. It is not present beyond the simple lobule. Here you can only see the A zone. The cortex of the entire caudal vermis, which projects to the vestigial nucleus, included in the A zone. So this is all the caudal part which is going to the vestigial nucleus. It is present in the A zone. These are the efferent connection of the cerebellar hemispheres. Then connections of the A zone of the vermis and flocculonodular lobe with the vestigial nucleus are indicated here. Here you can see the flocculonodular lobe and the vestigial nucleus. Then the B zone with the lateral vestibular nucleus. Then again you can see this the medial longitudinal fasciculus, which is present over here. and this is the medial longitudinal fasciculus which is present over here, and these are the third, fourth and sixth cranial nerve nuclei which are related to the medial longitudinal fasciculus and to the cerebellar hemispheres. So some vestibular fiber ascend bilaterally to oculomotor and nuclei and to the thalamus, and they also descends to the spinal cord as well, forming the spinocerebellar pathways. Forming the cerebellospinal pathways. These are the three peduncles, the superior cerebellar peduncle which is present over here, the inferior cerebellar peduncle which is present over here, and this is the middle cerebellar. Here also you can see inferior in this section, so over here you can see the inferior cerebellar peduncle which is going to the medulla, middle is going to the pons, and superior is going to the midbrain. So this is again another very important, and this is the summary of the today's lecture, the principal input and output of the cerebellum. So here you can memorize all of the lecture. First, the vestibulospinal, the spinocerebellum, both are the paleocerebellum in the vermis and the intermediate part, and the cerebrocerebellum, also called the neocerebellum. So the vestibulo is the oldest. This is also called archy cerebellum. It is anatomic region is the flocculonodular lobe. The principal input is from the vestibular labyrinth, and the nucleus is the lateral vestibular nucleus. The principal destination is the medial system axial motor neurons. Function is the axial control and the vestibular reflexes. Then is it mainly maintains the balance of the body. The spinocerebellum, also called the periocerebellum, which is related to the spinocerebellar pathway, both the anterior, the posterior, the rostral and the cunocerebellar pathways which are related to the hair. So anatomic region to these are the vermis and intermediate part of the hemisphere. Then the vermis, the principal input is the vestibular labyrinth, the proximal body part, the facial, visual and auditory inputs to the posterior lobe only. So this is going only to the posterior lobe. The nucleus is the vestigial nucleus, and the principal destination is the medial system is vestibular nucleus, the reticular formation and the motor cortex. And the function of this spinocerebellar system is to axial and proximal motor control and ongoing execution of the movement. Spinocerebellum, the intermediate part, is related to the spinal efferent, the distal body parts. Nucleus related to this is the interposed nucleus. The lateral systemic system, it is the lateral system. It is related to the red nucleus, that is the magnocellular part, and the distal region of the motor cortex. It controls the distal motor control and ongoing execution. So this one is the axial and the proximal motor control, and this one is the distal motor control. Next is the cerebrocerebellum, which is also called the neocerebellum. So it is related to lateral part of the hemisphere. The cortical efferents are mainly inputs going to the dentate nucleus, and the principal destination is the integration. The areas related are the red nucleus, the parvocellular part and the premotor cortex in the area 6. So it is initiation, planning and timing. So remember the input and the output of the cerebellum. This is very important. It is receiving input from the cerebellum, the cortico-pontocerebellar pathway about the motor activity of the cortex. Also it is receiving the input from the spinocerebellar system about the different control of the body, about the tone of the muscles. Also taking input from the vestibulocerebellum system about the balance, and it controls the efferent fibers going to the cerebral cortex via the dentatothalamic cortical pathway or dentatorobrothalamic pathway, dentatorobrothalamic pathway to the cortex. And these all interconnections, there are functions in a way that control a precise movement. So this very important function of the cerebellum is the precision of the movement which is necessary. So it is taking the input from the cerebral cortex, from the motor system, cerebellum and is controlling the cerebral cortex, giving information to the cerebral cortex about the precision of the movement. So these are the different input and output pathways of the cerebellum. And here I complete my today's class. Thank you very much for your attention. For the today's class, the references are the Carpenter Humans Neuroanatomy in the 9th edition, the Gray's Anatomy by the Susan Stranding 40th edition, and Netter's Clinical Anatomy. Thank you very much for your attention, and Allah Hafiz till the next class.